Hello, everyone, and welcome to today's webinar, Three-Dimensional Electron Microscopy of Brain Tissue Using Rapid Automated Preparation. My name is Stephen Goodman, and I'm the Chief Scientific Officer of Microscopy Innovations. I'm excited to be hosting this webinar and pleased to be able to introduce today's speaker, Emily Benson. I'll provide a more detailed introduction in a few minutes. Before we begin with the technical content, I'll provide some general info about the webinar and then about Microscopy Innovations. Yes, today's webinar will be available on demand after this live session via the Microscopy Innovations webpage under Resources, where you can also access related content such as the citations we referenced today. We'll make those easily available on the website. Next, we'd love to hear from you with any questions you have during today's presentation. You may send them to us using the question and answer button on the left side of your window. And type your questions in here and click Ask. We'll be answering the questions at the end of the webinar only. If you don't get to your question during today's webinar, we'll follow up with you afterwards. Last, we'd like to encourage you to share today's webinar with your colleagues via social media once it's posted online.
Thank you, Stephen, for that kind introduction and the opportunity to present some of my experiences and perspectives on using this automated tissue system and some of the work that we did to test it out. As a disclaimer, I'm obligated to mention that this is a research presentation and I'm not being compensated in any way. So both Renovo Neural and the Cleveland Clinic have had long involvement using serial block-based EM in academic neurosciences in academic settings and other specialties, and also commercial preclinical testing. This technology has proven extremely useful in both academic studies and preclinical pharma projects. I personally began using serial block-based EM as an undergrad. It was mostly just a case of being in the right place at the right time. I joined Bruce Trapp's lab in the beginning of 2011 when they had recently purchased a system for their lab, and I worked with Dr. Graham Kidd in getting that off the ground and developing applications and analyses to study myelin disruption on axons, axonal mitochondria, and smooth endoplasmic reticulum disruption in axons. And we looked at dendrites and their synapses and associated astrocytes. The Department of Neuroscience had a spin-off company, Renovo Neural, which invested in a serial block-based system about a year after Bruce Trapp's lab. And this led to me heading literally across the street where it was at, to work at Renovo on developing and implementing the assays and analysis developed in the Trapp lab for commercial usage there for pharma clients, industry clients, and then we also did academic projects for various people. As far as I know, Renovo was the first commercial group to offer serial block-based EM as a full commercial service. So between pharma jobs, we had the opportunity of investigating an enormous range of tissues and even, some materials at times. Our 3D EM unit was 2.5 people in a company of 20. And although I am only 5 feet tall, I was not the half person. It was actually Graham, who is 6 feet tall, was the half person. Also, Renovo's focus wasn't 3D EM, although that's what many people ended up knowing it for. But we were actually originally intended to be a small asset to the company to add on to their existing other myelin and axonal assays. Our goal was to obtain measurements that couldn't be easily obtained in any other way, which I will show an example a few slides from now about how we use this technology to measure 3D relationships in remyelinating central nervous system that otherwise would have been impossible to obtain. And that is like a strength of this technology for other areas as well. About the time that we were working on this collaborative project with Microscopy Innovations in summer of 2019, Renovo was winding down its operations, and Renovo Neural's 3D EM unit became the Lerner Research Institute's 3D EM Ultrastructural Imaging and Computation core. Graham Kidd and I, were fortunate to be able to continue our work as a subcore of the larger imaging core facilities that are run by Dr. Judy Drasba. And here we are able to continue to provide advanced large volume 3D EM imaging capabilities and to hopefully still contribute to the large volume EM community and provide services. We currently have 3 serial block-based systems. 2 are Gatan 3-view systems on Zeiss Sigmas, and we have 1 Thermo Fisher Scientific Teneo with a volume scope. So before I go further into what we're going to talk about today, I just want to give a quick explanation for anyone here who maybe hasn't worked with a serial block face SEM system. So in a nutshell, the sample is set up under a diamond knife in an SEM equipped with an in-chamber ultramicrotome. And then a section is cut from the sample surface with a diamond knife or a gallium ion beam if you have a FIB system. The fresh surface is then raster scanned with an electron beam, and an image is built up from the backscattered electrons to create an image that looks much like a TEM image. And then with repeated cycles of cutting and imaging, produce Z-stacks of serial images. So in a way, it's much like confocal microscopy, where you collect a Z-series, but here you're using an electron beam instead of a laser beam, and with 10 to 100 times better resolution. And with this technology, a strength is that you see everything there, whereas in other modalities, you sometimes, like for example, confocal, you only see what you stain for. So once you have a stack, it looks like, okay, there we go. So once you have a stack, there are a lot of different ways to use these. And here are two examples. Just to mention, like our main usage of data stacks isn't to create pretty pictures necessarily, but we enjoy it. But our real goal is to extract the quantitative data that you can't get through other imaging modalities. And serial block face EM provides large fields. They can be up to like millimeters long if you want. And 4 to 5 nanometer per pixel resolution. So this image on the left shows myelin in auto-segmented from a stack, and it's in blue. And then the right panel shows an oligodendrocyte in a single 2D slice here. And then, obviously from a 2D slice, you can't see everything going on. And then in this reconstructed image, you can see the oligodendrocyte, its associated myelin, which is in orange, and then, the axons are in white. Okay, so as I mentioned before, we use cellular block face EM at Renovo to measure relationships in remyelinating central nervous system tissue that would otherwise be impossible to get. And as we see here, the axons don't appear in one plane, because they undulate within the stack in the Z dimension and a little bit in X and Y. I mean, in a lot of tissues. This is corpus callosum, and they generally run in one XY direction, but in the Z, they undulate. It's like a bunch of spaghetti noodles. So in this example, we followed an axon down the midline in 3D, and then we extracted it from the stack so we could obtain myelin internode length, which is the distance between two nodes of Ranvier, which are shown here with the red circles, and then the myelin internode length would be this myelin sheath. In myelin research, longitudinal myelin growth is an important indicator of myelin recovery. However, quantifying it was rarely possible before this approach for using serial block face EM. And if anyone out there works on peripheral nerves, this can be thought of as a virtual teased fiber approach, because in peripheral nerves, since the axons are much larger and more robust, you can do this in real life, whereas here you do it virtually. Another application example of this technology would be mitochondrial analysis for endpoints such as size, volume, branching, Christie density, clustering, and more. Mitochondria and their Christie can be reconstructed. So in this image, mitochondria Christie are reconstructed from human cardiac muscle. And mitochondrial endpoints are really important to be, so they can often be used to show early changes in many disease models and are early indicators of disease recovery and the effects of therapeutics and other interventions. There are often semi-automated and fully automated analysis approaches utilized for obtaining mitochondrial endpoints, which, like all quantitative analysis approaches that utilize any automation, these require consistency of staining and tissue quality, which leads us into the next area. So a key constraint for serial block face EM is sample preparation. And while staining is mundane, boring, and tedious, and sometimes very mentally painful when you have so many other things to do, but you have to stand around for hours and move hazardous liquids around with disposable pipetters. But despite this, many people are greatly interested in sample prep, mostly to work towards reducing the challenges and bottlenecks that arise from it. So in the slides before this, I highlighted a few applications that many labs routinely do to highlight that serial block face EM is an important technology to glean data that are difficult or impossible to obtain in other ways. And serial block-based EM is a relatively easy technology to use, as in the serial block-based systems and the software themselves. So it often seems a bit ironic that one of the most challenging parts has been the low-tech sample preparation that has existed throughout the years, because the staining isn't actually difficult. It's just really labor intensive when you do it in a manual way. It's time consuming and you're handling toxic chemicals for hours. So we were very excited to investigate an automated approach to sample staining and embedding, which reduces these factors potentially, which we looked into, and especially the human time factor. And at Renovo, with the standard manual staining we would do, we increased the time on certain steps, which made the process even longer and more painful. But as a company and now as a core, we care a lot about having good, consistent staining, which opens the doors to easier auto segmentation and AI approaches, such as deep learning approaches, which was important to us as a company who was doing serial block-based EM studies for pharma and commercial companies as a high throughput approach to mine quantitative data from serial block-based sets. And I say high throughput with like finger quotes in the air because it's still electron microscopy, so you know how that goes. Um, so while there are some other staining systems to accelerate some of the aspects of staining, we had resisted them due to concern that they might alter the tissue in ways that would make trouble for us. And some other automated or semi-automated staining approaches we had seen seemed like they would have been, like the systems would be forever contaminated if we put heavy metals in there, and that would render them impossible to be used for other processes or for people who want to use the systems in other ways, like for immunostaining or other things. Also, most importantly, for us, automated staining approaches they weren't the idea of that isn't of much use if the results are not comparable to the best manual staining. If, like, for example, there were more shrinkage of the tissue or another effect on tissue so that ultrastructural measurements were not comparable to older data or existing literature, that would be a deal breaker for us. Likewise, if the staining density or staining penetration were so different that images couldn't be handled by our existing image processing pipelines for analysis, then the automation of staining would not be useful. I guess overall, we didn't want to reduce the time up front if it were just going to break our workflows downstream and not end up in a net time saving situation. Especially as a company, and even now as a core, we had clients who would return year after year from both academic labs and commercial companies. So we couldn't introduce automated staining approaches or any new methods that would render their historical data useless. So as an example, drug companies that maybe wanted to test a new compound, but they'd worked with us other times in the past, they often wanted to use their historical control or naive groups, the data from those from years prior that we had worked on. But if we changed up the staining protocols to a method that caused ultrastructural changes to the tissue post hoc, then we'd be in a world of trouble when comparing their historical data to their, more recent additional added groups, since you wouldn't know where the changes actually came from. Also, there were a lot of academic labs who had multi-year long research grants and, long longitudinal studies in some cases. So we might have gotten, some of their samples to run by serial block-based one year, others another year, then some of the transgenic animals a different year, or however it worked out. And in some cases, project involved human donor tissue, and these labs might send a few tissues each year, depending on when it became available. So basically, if we had drastically changed any of our methods for a staining approach just to save some time, but if it would render their previous data useless when compared to, analyzing the data after such a change, that would be basically awful for everyone and a huge waste of research dollars and render the experiments unable to be, completed. So we wondered if microscopy innovation system might speed up the processing, yet bypass all the concerns that we were worried about. So we set up a study to give it a try. And in this collaborative study, we investigated whether samples stained using an ASP1000 automated robot system provided comparable data to our in-house manual staining. So the tissue, we used rats, and these were perfused with a modified Karnofsky's fixative, 2.5% glute, 4% PFA, and a 0.1 molar sodium catalate buffer, but people often use phosphate buffer too. Either is fine. We stained rat brain, cortex, corpus callosum, spinal cord, liver, and kidney samples. We went on to analyze the cortex tissue in detail and examined axons, myelin, synapses, and mitochondria from both manually stained cortex tissue and cortex tissue stained with the ASP1000. We chose to look at these endpoints because those ultrastructural features are often used as study metrics in preclinical studies in many research projects, and you can see small changes within these if they would have occurred due to staining. The cortex tissue used in these analyses to compare staining modalities was from the same animal, so it was a within animal analysis just to be sure that we didn't have differences based on animals. The samples were imaged on the Thermo Fisher Tenio volume scope in high vac at 2 kV, and if anyone's interested in the exact settings, I can share that further. So we'll start with the beginning, where how to use this. We use the system, how we loaded the tissues into the ASP1000 system. This diagram from Microscopy Innovation shows loading the tissue into an MPrep capsule. We use these MPrep capsules for the cortical tissues. The screen is held in place by the insertion tool, and then the tissue is set on the screen. And then you encapsulate the specimen with the other part of the capsule. Then you push the screen into place with the insertion tool, and then you load it onto the pipetter here. And so the staining is conducted in the ASP1000 system shown here, and the staining is robotically handled. The user does a little bit of upfront work, loading their tissues, making up their reagents and putting them in the well plates, and this arm has the ability to move in XYZ. As Stephen mentioned, the system is fully enclosed. It vents into your hood, or the user can choose to put the entire system in their hood or remove the casing if it doesn't fit in the hood with the casing. So there's a lot of flexibility. Reagents are loaded into the 96 well plates, or optional trough well plates, or other plates depending on the user's usage of the system. So the plates can be covered and sealed with an aluminum sheet or a plastic sheet, or unsealed if it's not something that's going to evaporate quickly. 6 plates can snap into the base of the system, and from the computer is where the operator creates and or alters the protocol, and then that can be saved and repeatedly used each time, and other users can run the protocol, which reduces variability between people, and it just makes sure that everyone's using the same protocol. And the operator can choose to have the robot send texts or emails, and there's also an LED light, and you can have it change to different colors to mean different things in your workflow. So you can just look at from far away and see the status, or you can have it send you texts or emails. It's really adjustable based on your needs. When I was trying out the system, I played around with the organization of my solutions and plates to make cleanup easier. For example, like a user could choose to put all things that are very hazardous in one plate. Then a user could put all the things that are not hazardous and able to be dumped down a sink into another, or any other variety of organization, the way they want to organize their things based on the workflow and their needs. So that does make cleanup easier, handling the used reagents easier. So importantly, the automated processing using the ASP1000 reduced the staining time from about 1 week to 1 day, and the operator effort from about 2 days down to about an hour. So just kind of going through the recipe. So the solutions you make are the same between the manual and the automated protocol, at least in this case. A user can take a protocol that is used for their manual staining and easily replicate it and apply it for usage within the ASP1000 system. And this is a pretty standard serial block-face EM protocol that was used for many of the tissues shown in the next slides. And there was some tweaking in some steps over time, but nothing majorly deviating from this. I may have had about 4 or 5 versions of the protocol saved on the Microscopy Innovations laptop by the time I was done with it. But it was just like small things. It's very customizable based on your needs and personal workflow. Also, it's important that you can have some of your tissues go through a certain step while others don't, for example. although we rarely use tannic acid, we just wanted to try out this capability. So with a couple tissues, we had them go through tannic, while others were just in the buffer, while at the same time, because in your 96 well plate, you can have a row that maybe has 2 wells with tannic, and maybe that's lined up with the capsules that are number 1 and 2, and then the others in that row would just be getting buffer at that time. So this is great for other approaches too, like if you want to optimize a protocol or optimize something for immuno EM, or you want to have different concentrations of antibodies in different wells, or other experiments in protocol development where you can do it all in one day instead of having to have a graduate student or a tech be working on um, protocol development that doesn't need to take as long. So to just run through some of the steps in this diagram, the tissues were stored in Karnofsky's after perfusion and to be well fixed. And then just before we were going to stain, we transferred them to cacodylate buffer, and then we rinsed many times in cacodylate. And so this is where they would have actually been loaded in the capsules, and then they're put on the pipetter, and this is where they begin with going through the ASP 1000 system. So here we just rinse many times in cacodylate buffer to remove any aldehydes and phosphate before going into the reduced osmium, which is chilled, and then we put it in just before we need to use it. After the water rinses were complete, at this point I had the ASP 1000 send me a text and also do what it calls an aspirate and hold, where it sucks in or aspirates liquid from the well and then just holds that in the capsules, so that they stay wet. And in this case, I did that when it was on the final water rinse, so that I could this allowed me to add the 60 degrees Celsius TCH that was freshly made up and still at 60 degrees Celsius. The version of the ASP 1000 that I was using did not have the temperature control platform, but there are ways around that. And there's also a new version of the ASP 1000 that does have the temperature control, which does add additional functionality and reduces user interaction even further. I also did the same aspirate and hold process for the last water wash before the let aspartate step, so that I could add in 60 degrees Celsius Walton's let aspartate when I got to that step. Everything else, is pretty much the same. And then, you go through, then you wash again after lead, you go through your graded ethanols. We used acetone instead of, we usually use propylene oxide, but, you don't, I didn't feel safe using that also because I didn't try it out in these plastic plates. Some plastic is okay when you use it, but acetone works fine as well. Then we did a mix of epoxy and acetone, and we did 100% epoxy. And this just says epoxy here, since people should use whatever they prefer and whatever works best for their lab. But in this study, I used the EPON 812 substitute from EMS, and we use a medium hard recipe. This is our routine resin for everything. It works best in our hands with our tissues. But I know people have strong opinions about others, and that's, whatever works for you is awesome. And, and that's the thing about this, you can modify it for what your lab does. So as we mentioned before, speeding things up is great, but only if it's not at the expense of ultrastructure, and it has to not disturb the morphological features. So here are some of the results, starting with staining penetration. So tissue staining quality was comparable between methods. Here is a cube to illustrate staining penetration, which appeared to be very even throughout the tissue. This shows so this is overall like a small region through the image area of a piece of cortex that was manually stained versus in the ASP 1000. They're both well stained, nicely contrasted features. The membranes appear intact and nice, and it appears even throughout this tissue sample. And these blocks are representative of the much larger sample that was used for this project. Sorry, my slides are not moving. Okay, there we go. Also, we looked at how the ultrastructural features look between methods. We were able to resolve critical smaller ultrastructural features such as mitochondrial cristae, synaptic vesicles, and membranes looked nice, synapses looked nice, cellular membranes appeared well and intact, and cytoskeletal features also look uniform and haven't been disturbed post hoc during tissue handling. Also, we did not see any heavy metal precipitates that we have seen when people did inadequate rinsing or washing and manual staining methods. The ultrastructure, oh, there we go. The ultrastructure looked good. Membranes were well contrasted, and postsynaptic density details were clear and well resolved. So the top graphic here shows a dendrite with three spine. Well, okay, so with three synapses. Actually, not all. There's not three spine strings. Sorry, there's just three synapses. And we've had a lot of experience with synapse reconstruction, and so any deficits in staining or embedding become limiting when doing this sort of analysis we found. So, you know, here we just wanted to check and see how the tissue looked, and the vesicles look well resolved. The details were clear and well resolved. It looks great to us. And so moving on, I'm not sure what's, okay, there we go. So here is a reconstructed presynaptic terminal showing mitochondria with convoluted shapes. So it's an inhibitory axodendritic synapse, and these are pretty hard to spot if your staining isn't good or fixation isn't good. In addition, trying to figure out mitochondrial organization in poorly stained tissue is very difficult, partially because poorly stained tissue has issues with cutting, charging, and thus forcing a user to use thicker cuts at times or having skipping, thus making it hard to catch fine ultrastructure or like odd mitochondrial shapes if they have thick slices that you're not gonna be able to see all these. You might miss something because you're gonna have a lot of interpolated space between sections. On the next slide, we're just so for us at Renovo at this time, and then even now we still do myelin analyses, but we do a lot of myelin analyses still. One important measure of myelin is the G ratio, which is the ratio between the axon diameter and the myelin's outer diameter. And this is an important study in animal models of demyelination and in human disease, and preclinical studies for drugs or compounds that promote remyelination often look at G ratio, which we could measure in conjunction with internodal length, which I showed before, which was cool to have the G ratio and specific internodes they came from data together. Okay, so this G ratio analysis involves a pipeline that has automatic segmentation and measurement. And if the myelin had significant defects or structural deficits resulting from tissue handling, that would have broken the pipeline. But that didn't happen. The tissue looked great, and to check for any minor changes, we my slides are kind of lagging when I go move forward. Sorry about that. We compared the G ratios in the manual stain versus ASP stained cortex from the same animal. So we looked at axon diameters, myelin thickness, and G ratios, and the values were similar between the staining methods. This was a good indicator for us that the ASP 1000 didn't cause any shrinkage, expansion, or rough up the tissue in ways that would have affected important ultrastructure and the resulting quantification. So with all these examples, what we wanted to illustrate was that the staining was consistent, it had good contrast, and it didn't affect the tissue negatively or in a way that would render early data unusable, which had been a major deal breaker for us. And as I mentioned in an earlier slide, having good consistent staining opens the doors to easier auto segmentation and to AI and deep learning approaches that are used pretty regularly now. People are starting to apply. So the final test we did was to compare tissues stained by the ASP 1000 versus manual staining to see if a convolutional neural network that had been trained on manually stained prepared tissue would also work on the tissue that we stained with the ASP 1000, because that would mean that the tissues were so similar enough to do this, and that labs could potentially switch staining methods to save time without having to retool all of their workflows or break their existing pipelines. And we used a UNet network, which is a very popular convolutional neural network approach for medical imaging. And we did this out of Dragonfly. And it was very, very quickly trained on only 3 slices of data. And although there are some bits that need to be improved, it was the same for both and not related to the staining. So, for example, since this was a quick example, you can see that it picked up some features in the nucleus, but that would be easy for it to initially initially train it to find your nuclei and then mask them out and then do the segmentation and quantification. Also, like simply training it on more than 3 slices would get rid of most of the issues. But we were just trying to actually see how badly could you do this and still get like, uh, you know, is it still going to work? So here, the Golgi apparatus membranes are in magenta. The mitochondrial network is shown in red, and the smooth endoplasmic reticulum network is shown in green. Except for just ignore the nucleus part. We talked about that. All right, so I'm just moving on to the next slide. It's just kind of stuck. Sorry about that. Okay, so Graham made this video. Graham Kidd, who worked with me, he's the director of our unit. And so I just wanted to show this because it was pretty. And so here's this little video, and then we'll move on to the next slide. And he made this in Dragonfly. We've been trying that out, and we enjoy it so far. So just real quick, here's a quick image of some liver from manual staining and ASP1000. One image has a bit of an issue because the scope developed a bit of an issue. It wasn't the sample, but here in liver hepatocytes, mitochondrial structure was comparable. Glycogen granules were prominent in both tissues, and endoplasmic reticulum was also well labeled. So as a conclusion, multiple conclusions. This was a very limited initial study, and there was no opportunity to extend it at Renovo Neural. But with what we worked on, it was adequate to show the benefits of this automated staining approach and that it could be used without affecting earlier projects or causing major problems within a lab's workflow. There are also other tissues we ran out with the ASP1000, and some we can't show because they were client samples, specifically the spinal cord samples, but they looked great, and perhaps we can soon extend the analysis to some of the other tissues on hand that have been stained and embedded with the ASP1000. Using the ASP1000 sped up the staining tenfold at least, and there were no obvious qualitative or quantitative differences. Programming the system, when people are setting up their protocol within the ASP1000 software, it allows a lot of adaptability to different staining requirements, such as plants, non-mammalian model organisms, immunostaining, basically any staining you do in your lab normally. The safety aspects were really well handled. I really worry about safety things, so I felt pretty good about this, which Graham kind of makes jokes about that if I feel okay with something, then it's probably okay. So it ducts to your hood, and then with attached before along the duct that goes from the system to your hood is like a vacuum fan type thing. I won't call it a vacuum. It's like a fan. I can let them talk about that. Let the microscopy innovation people talk about that. But yeah, so it pushes the air from that into your sucks the air from the ASP1000 into the hood. Or you can put the system in the fume hood. So you can do small batches, only 8 capsules at a time, but you can actually still do more in a week than if you did, a manual batch of 20 samples. Plus, you have all your time to do other things while the robot stains for you. Because even if you're doing 8 capsules at a time, if you start a staining batch every day or every other day, because your samples that you start on Monday, they're going to be out of the ASP pretty quickly, and then while they're curing in the hood, while they're curing in the oven, you can start another 8. So it's actually really nice, and you have all your time to do other things instead of having to stand there moving around hazardous liquids when you do manual staining. There are also endless possibilities for modifying and introducing other steps or trying alongside other samples. So that kind of wraps things up. And on a less serious, I'm just being a little bit silly, it's kind of very important. The ASP1000's text and email capabilities can be used to boost your employees' morale. When it had to text me to tell me what to do or give me an update of where it was up to, or I would have it text me to tell me that it's going to soon be at the TCH step, so I would make sure I have it all finished being made. So when it gets to that step, it sends me another text and it tells me I have to put the TCH in. Well, I didn't really want to be told what to do by a robot, so I would have it sign its texts with love ASP1000 or other kind messages. Also, since it's 2020, you may not be able to be social with people in your lab. But if you know they're about to use the ASP1000 system tomorrow, you can edit the protocol they're about to use and add links in the texts or the emails that, the link can go to a picture of their favorite animal to show that you still care about them and you're thinking about them. Because the world needs more kindness, even if it comes from a robot. So thanks for tuning in. I just wanted some acknowledgements. I want to thank everyone at Microscopy Innovations, Stephen Goodman, Tom Strader, Mark Nelson. They've all been really helpful during this whole process. And I wanted to thank Jolene. She helped immensely and gave really helpful feedback. And I wanted to thank Graham Kidd and Deandra Starks, who, they work with me. They're great. I miss, I see Graham almost every day, but, we had a great time, 3D team for life. And then I wanted to thank Jay Campbell, who kindly stained some of our tissues for us using the ASP1000 system. And, we sent him our protocol, and, he checked to see if he could, integrate it with what they had already been doing. So he did that before we had a chance to try it out ourselves. And so.
Hi, Emily. This is
Oh,
Tom.
hi.
Can you hear me?
Yeah, hi. Sorry.
We can, I think, go into questions now, and I can read the first one. It's from Marcella at West Virginia. Can you explain why the process are speeded up with the instrument?
Yeah, so this works because the ASP1000 continuously flows reagent past the specimen. It aspirates and the way it moves the reagent around your tissue constantly. So it helps with speed up diffusion into your tissue. And Tom and Stephen at Microscopy Innovations could explain this further if they would like to. But the reagent, moves across the tissue every second or two. And for example, in the protocol in the M&M poster, the osmium ferrocyanide, reduced osmium flow to the specimen, every 2 seconds within an hour. So it does like a total of like 1,800 passes around it. So it's the continuous mixing and movement of the reagent into, which helps it to diffuse into the tissue, from my understanding, unless Microscopy Innovations would like to add more.
No, that's pretty much it. I think also being able to go right to the next step when the step you're on's done. When the human's doing it, the timer goes off and you might be busy and you're not there at exactly, the optimized time to go to the next step.
Right, yeah.
Also that. But okay, thank you.
Sorry.
Moving on. Yeah.
Yes. Hey, Tom. Thanks so much, Tom. Tom, while you're on, this is Steve Goodman here. A question came in asking about heating stage, heating and cooling. Can you answer that? I think that's probably a better question since Emily mentioned it.
You betcha. What exactly
the question was, can you heat up the plates for processes when the chemical solution needs to be used warm?
Absolutely. Yeah. There are two of the stations have been, you can buy a thing called a TCU, a temperature control unit, and add two positions that are heated or cooled. It will go down to 0 or up to about 90. But 60 is where the target, of course, if we're doing polymerization. And then, as Emily mentioned, for the TCH, it is warmed in many cases when it's prepared, and then also when it's used. And then when experiments were done at UW-Madison and then at Stowers Institute, also the dehydration was cooled.
Great. Thank you, Tom. So, Emily, there were several questions about the protocol. Can you move to the protocol slide?
Yeah, I'll go back to that.
So while you're doing that, one question asked, how is the preparation speed so fast? And this a second question, which I think they may be answered together, was, can you explain what the automated exchanges column means in the green automated column?
Oh, okay. So this is showing the manual steps, like how many exchanges we do. For example, I do 6. So in the manual side, I do 6 washes with clean buffer. Here, I did, I had 3 rows of it, and, so with the 30, 30, 30, what it means is, like, so it did the, aspirate and then, release the over, like, 30 times over the tissue. So, but it's still from, like, the same, row of it, right? So I have 3 rows of clean buffer, and it does, you know, and when it goes to, row 1, let's say, it does 30, like, sucks in and releases the clean buffer, and then, moves to the next, row that has clean buffer, because I'm trying to get it, like, more and more clean, with each time I, do a exchange with the new buffer, if that kind of helps to explain it. And then and so, with, so for example, after this, second osmium here, you wash in water a lot of times before you go into the urinal. And so this is basically showing the same thing. So with a manual, you do, 5 washes with water. And then here, this shows that there are 5 rows of it. So, and then each time it's in the new row of water, it's gonna, lift up and let go of that water 90 times. But each of this 90, 90, 90 indicates a row of clean water.
What Emily's explaining is that basically why it's so fast is that we have 90 exchanges of the reagent sitting in each well, for example, on the water one. So there's a lot of exchanges. That happens just over 15 minutes. So there's very efficient rinsing. Another question was about osmium tetroxide. Is there a way to prevent CO2 precipitation?
Is there a way to prevent, sorry, on which step? Sorry.
On the osmium step.
Oh, on the osmium step, is there a way to prevent precipitation? We haven't really had any problems with that. I guess we just haven't had any problems with that. I mean, if it's on your surface of your tissue, I guess I wouldn't know. So if you're doing something with grids, if something changes, but I haven't had any precipitation problems. I think that has a lot to do with washing it well before and after the osmium steps.
Yes. Okay.
Oh, wait. Graham mentioned did someone mean lead rather than the osmium? Wait.
The question asked about osmium.
About osmium?
Yeah, but maybe they meant lead.
Okay, it'd be the same kind of answer. It just matters well about washing. Oh, and with lead aspartate, it's not the same as, like, so the lead that's used for TEM is different, is lead citrate. This is lead nitrate mixed with aspartic acid. So lead nitrate doesn't have the same issues that people run into with lead citrate. But I know there are ways around that because people use this with grids for TEM, but I haven't actually done that myself, so I don't want to mention about lead citrate when I've only worked with lead nitrate with this system. But I know Stephen has a lot, but a lot has to do with being made fresh. But I know Stephen has a lot of experience with using the lead citrate.
Okay, great. There's a very specific question about the G ratio measurement. The question asked, you mentioned N equals 1. Is that 1 axon or 1 block?
That's
Can you explain?
1 animal. So, because we did it within animal experiment. Let me go to the slide too. We did it within animal experiment. So I think we did like we did like probably like. Oh, sorry. I think I went too far back. I'm so sorry. Sorry about that. It was 100 axons and like the same animal. Like we didn't do like, because we did it with one within animal experiment, but we could expand it to, but because it answered the question with having 1 animal, because that's really what we wanted to know. Like, does using the system change anything between this 1 animal where it's like 1 slice of tissue versus the next, chunk of tissue that we stained up? But I'm still going to go to it, but sorry about that. I think it's the next slide. Sorry. Okay, so what we meant by N equals 1 is that this is like 1 animal. We would need to do like multiple within animal, things maybe, or like, but for what we were aiming for, we didn't need more than 1 animal. But
so about how many measurements was that?
100 axons, so 100 G ratio measurements.
Per animal.
Yeah, or maybe, okay. Could have been more or less depending on, I can pull up the original data and look at that but yeah, it would be a lot, like definitely between 70 and 100 per animal.
Yep.
But per staining condition, I mean.
Right.
Sorry about that.
Okay, great. So another question was, will the specimen preparation methods and protocols work for FibSem?
Yeah, because the same staining procedure is used for both ways of doing serial block face. So people do the same protocol. People use the same type of protocol for fib versus diamond knife cutting. So yeah, the same should work.
Okay, great. I think the last question, actually the last two questions, did you have any downtime or reliability issues with the system? We may also want to bring Tom in on that as well. Tom is the VP of product development for Microscopy Innovations, and we heard from Tom a few moments ago.
I didn't have any issues like that when I was working with it. Sorry. There was no downtime or any issues that I ran into.
Yep. Tom, do you have anything to add?
Nope.
Okay. Great. Let's see. Did you have any problems with charging?
No, I don't usually have any problems with charging because we increased. So with our manual staining, we increased the time of urinal a lot longer than most people, and that seems to get rid of most problems. And I also do a higher concentration of lead than most people. And I don't really have charging problems really ever. That has a lot to do with a variety of different parameters, though. Some of them are going to be your imaging parameters. Some of them are going to be your tissue, your resin, and how well. So another thing is, like, besides the staining, so how well was it perfused? How well was it fixed? But generally, if someone, I don't really have problems with charging. And like, if someone is having problems with charging, I'd be willing to help them troubleshoot that and try to figure out what's going on. Sometimes you can tell by just looking at the tissue what's causing it, and by looking at what membranes are well stained versus what aren't, because then that can give you a hint of maybe what staining step needs to be done longer. But also, so people could have issues if they're looking at tissues from other areas, like something that doesn't take up very much heavy metal. Like if it's a very collagenous tissue, that way might be a different story. But there are ways you could stain collagen, even though it's like a protein. So like heavy metal, it might need to be a little bit different. But there are ways around charging issues. We'd be happy to help if we were able to.
Excellent. So we're kind of running out of time. There's one last question that that I'm going to ask from the list, and then I think we'll close it out. But we'll address everybody's questions via email later. How much reagent did you use compared to your manual protocol?
Much less, because I usually use dram vials, which I have to put at least like 2 mils in, whereas in these 96 wells, I put in. I'm just looking where I set my notebook about how much I use, but Tom Strader can also tell you exactly how much to put in there, because you put a very specific amount in the 96 well, depending on also like which capsule type you're using. But Tom can tell you about how much reagent goes in each type of well plate, because like the trough needs a certain amount, the 96 wells need a certain amount, but it's much less than I would use if I were using the glass dram vials, which is also really helpful for people who are doing like immunostaining and using expensive antibodies and such. But Tom can tell you the exact amount, because I have my notebook with that information, but it's not right in front of me.
Yeah, right. In the 96 well plate, it can be as little as a half a mil per well, per channel. And then a mil is what we typically use. In the trough, which is a column, really, it's a 12 column plate. They hold 7, and we put typically 5. Again, if you want to make sure you're conserving reagent, you put smaller, and then if you want to make sure you've got plenty for a rinse or something, you want a larger volume. One of the good things about it, though, as Emily mentioned, if you want to do 2 or 3 runs in a row, you're not taking much risk by reusing a lot of the reagents, because it puts them right back where they came from. The first rinse after osmium, that one you might want to swap out. And even at that level, you just can replace the reagents right in the well of the plate for the next run, which is fairly easy.
Yeah, that's great. We are running up against the hour. I want to thank everybody for attending. I want to give Emily a round of applause, obviously virtually, for providing us with this very informative webinar. We'll follow up with any questions we weren't able to answer. Thanks again for attending. Have a wonderful day. And I also want to let people to know on our website, Emily's publication, including the poster, which some of this work that she described is described there, will be available or is available on our website. Thank you all so much for attending, and have a wonderful, wonderful rest of your day.